very much for this alluring introduction, Amanji. Uh, मेरी वॉइस ऑडिबल है? हेलो?
Sir, you have to wait for 5 minutes as there is some technical issue in YouTube on streaming.
ओके. हाँ जी, आवाज आ रही है. हाँ, आ रही है. अच्छा, हाँ, ये शेयर हो ही नहीं रहा. मैं मेल पे जाके किया वैसे. ठीक है. हाँ जी, हाँ जी.
Yes, sir, your screen is visible, but we have to wait for 5 minutes to resolve that sound issues in YouTube. I would like to remind all the participants that the feedback form will be shared after the end of session. An e-certificate will be provided to all the students who will fill the feedback form. So please, all the students surely fill the feedback form. Uh, Now, I would like to call Dr. Harminder Singh to please deliver his talk. Sir, please. Sir, please unmute yourself.
Is it audible now?
Yes, sir, you are audible now.
Good morning, everybody. Uh, Chancellor, of university, pro chancellor, pro vice chancellor, registrar, uh, managing director, and convener of the event, Dr. Amit Sharma, Dr. Minakshi Sharma, organizing secretary, and all the listeners. Uh, First of all, I would like to congratulate you all for organizing this such a wonderful and informative seminar. And I also thank you for giving me opportunity to deliver my lecture in this platform. So thank you very much. So today we'll discuss about the topic. The topic is cell phone radiations and human health. Uh, Aman, my voice is audible to you?
Yes, sir, you are audible.
Okay, okay, thank you. So today we'll discuss about the cell phone radiations and human health. We are surrounded by different types of radiations. Some are harmful, some are non-harmful, and recently it has been reported that the cell phone radiations they operate in a cell phone. They work in a radio frequency radiations which are not harmful to the human being and other organisms. But the exposure level becomes so intense. It has been reported that several health ailments, including cancer, have been reported due to exposure to these radiations. So it becomes very necessary to discuss about the cell phone and their effect on human health. That's why I have chosen this topic. So let's we start. Mobile phone may be called the fourth basic need of our life. Mobile phones are used in every sphere of life due to which the user are at high risk of exposure to electromagnetic fields radiated by these devices. So if we talk about electromagnetic fields, electromagnetic fields are invisible line of force that occurs whenever electricity is being conducted. These forces occur from both natural sources such as sun or atmospheric sources and solar disturbances, and from man-made sources like electric lightning, microwaves, television, cell phone and computer, etc. So there are two types of electromagnetic field radiations. First is the ionizing radiations, second is a non-ionizing radiations. You can see in the electromagnetic spectrum the ionizing radiations, X-rays, gamma rays. These are the ionizing radiation on right hand side. They have a low wavelength and high frequency. Non-ionizing radiations include ultraviolet rays, visible light, infrared radiation, microwave radiations, extremely low frequency radiations. On the right side, they have a high wavelength and low frequency. If we talk about ionizing radiations, these include gamma rays, X-rays, alpha particles, beta particles, and neutrons. The energy level of ionizing radiation is high enough to destroy the bond present between the atoms and molecules of the body cell. This is proved beyond any doubt that ionizing radiations are associated with a lot of health ailments, including cancer. What We are not exposed to ionizing radiations uh, during a lifetime. Once in a while, we exposed to these radiations. Moreover, when we have to exposure during X-rays and other procedures, medical procedures, we take necessary precautions. If we talk about non-ionizing radiations, these are described as a series of energy waves composed of oscillating electric and magnetic fields traveling at a speed of light. Non-ionizing radiations include spectrum of ultraviolet, visible light, infrared, microwave, radio frequency, and extremely low frequency radiations. All electrical appliances and electronic devices produced damaging electromagnetic fields which are harmful to biological systems. So these are the different sources of electromagnetic radiations: microwaves, electrical lightning, telephone, laptops, computer, and so many other things. All the electric and electronic gadgets uh, in a domestic environment and workplace, they emit these non-ionizing radiation. May it be microwave radiation, they may be infrared radiations. So non-ionizing radiations a matter of concern, although the non-ionizing radiations are considered safe because its energy level is not enough to destroy the bond between the atoms and molecules of the body. What excessive exposure to non-ionizing radiations may affect biological tissues through other mechanisms. Due to evolution of mobile phones, we are exposed to non-ionizing radiation 24/7. These are some recently research reports gathered by National Toxicological Program. You can see here, these are the results showing clear evidences. Several studies have been reported that non-ionizing radiations leads to cancer. On the other hand, you can see here in the green color, uh, these are the studies which show no relation between the non-ionizing radiation exposure and the incidence of cancer. And rest is there are some evidences, there are some equivocal evidences. In nutshell, you can say the non-ionizing radiations, they are a greater concern to the human being because we are always being surrounded by these radiations. So on the basis of detailed review of the literature produced by some world organizations as Environment Protection Agency, National Institute of Environmental Health, World Health Organization, National Radiological Protection Board, International Radiation Protection Association, the Scientific Committee on Emerging and Newly Identified Health Risk, and some other research reports. The International Agency for Research on Cancer in 2011 confirmed the radio frequency radiations, which is a form of non-ionizing radiations of the mobile phone, as a possible carcinogen, a hazard for human health, and classified them in Group 2B in 2013. ग्रुप 2B में उस तरह के सब्सटेंसेस आते हैं जो कि जिसकी पॉसिबिलिटी है कि वो कैंसर कर सकते हैं. So it means that radiations emitted from the mobile phones, they may be carcinogenic. So let us have some watch on the other electronic gadgets uh, and their harmful effect on the body. Computer. If you talk about the computer, the electromagnetic field emitted from the computer monitor have been reported to cause different types of abnormalities such as change in the blood pressure, headaches, and skin disorders. The cause of potential risk of adverse pregnancy outcomes and such as miscarriages have also been associated with the effect of electromagnetic fields. Then comes microwave oven. The study found that the broccoli, when microwaved with a little bit of water, loses up to 97% of its beneficial in antioxidants. Some some amount of phenolic compounds and gluconisolates were also lost. The study was published in November 2003 issue of Journal of the Science of Food and Agriculture. Another study found 60 seconds of the microwave waving a garlic depleted the food of its alliinase, garlic's primary cancer-fighting ingredient. The microwave heating turned 30 to 40% of the vitamin B12 in the milk into a dead form, which was found in Japanese study. Then microwave exposure adversely affect the male fertility also. Then comes mobile phone towers. They also play a damaging effect on the health of the human being. During the last 5 years of these researches, it was found that those people living in the vicinity of 400 meters of the cell phone tower had reported cases of breast cancer, topped the list. What cancers of the prostate, pancreas, ball, skin melanoma, lung and blood cancer were all increased. So what are the theories how these non-ionizing radiations they affect the living system? So several theories have been proposed time to time. One theory is that the electromagnetic radiations of certain frequencies make alteration in the T lymphocytes and other immune system leading to immunosuppression, partially through the induced calcium ion efflux. And another theory advocated that electromagnetic field affect cell growth and reproduction, while third group hold the view that EMF make the cell manufacture proteins which they normally do not produce. And another theory reported that paramagnetic properties of iron storing organs like spleen, liver, and blood, uh, you can say bone marrow, make these organs more likely to be affected by the magnetic fields. In this regard, I have conducted a study. You can see this is a norm, normal scanning electron microscope micrograph of blood of Balsi mice. It was removed and taken in a wheel. The experiment was performed in vitro. The blood in a wheel was exposed to cell phone radiation for 2 hours. After that, you can see here, this is the case of red blood cells. Most of the red blood cells they show poikilocytosis, means they have abnormal shape, and isocytosis and macrocytosis. So it shows the effect of magnetic field on the iron containing tissues in the body. Now let us come to the cell phone radiations, how they affect our body. Mobile phone nowadays not only used for its basic applications, but as a means of entertainment and spending leisure time, due to which the user are at high risk of exposure to these electromagnetic radiations. Mobile phone is one of the fastest growing technological developments, becoming an inseparable and necessary part in modern life. Today, more than 5 billion people, 67 of world population, are using mobile phone worldwide. Number of smartphone users in India alone reached over 760 million in 2021, while the number of smartphone users exceed over 3.8 billion by 2021. Radiation from the mobile phone pass invisibly through the human body and interfere with the body's biochemistry. Uh, Earlier, we also studied that International Agency for Research on the Cancer in 2011 confirmed that the radio frequency radiation from the mobile phone as a possible carcinogen hazard for human health and classified into Group 2B in 2013. The smart mobile phone, they are uh, because of our excessive dependency on these mobile phone, they are very harmful to us because we are exposed to 24 by 7. We also get the passive exposure. We have a great dependency on these phones. So smart mobile phone, they emit more radiation as compared to the basic phones, because the smart mobile phone they have a series of antennas in the handset that can receive and transmit from multiple sources. Latest generation cell phone have a capacity to switch automatically among the various frequencies due to multiple input and multiple output system. So you can see this is a structure of a mobile internal structure. You can see different types of antenna here. This is a Wi-Fi antenna, and then GPS antenna is here. Means in a mobile phone you can get the different sources, different places where the emission of radiations happen. This is the structure of a MIMO, multiple input, multiple output. This is a mobile phone receiver. It has a multiple antennas, antenna 1, antenna 2, antenna 3, antenna 4. So these all antennas, they try to receive the radiations from the surrounding towers, due to which just to increase the speed and performance, the latest mobile phones, they are becoming more and more source of radiation emission. Now, if you see the mobile phone, how they affect the living organism, mobile phone affect the living organism by two mechanisms. One is a thermal effect, second is a non-thermal effect. You just see here in the image, this is a thermographic image of the head with no exposure to the harmful cell phone radiations. And second image, you can see the red area in the head, which is the thermographic image of head after 15 minutes of cell phone exposure. So what are the thermal effect? If we talk about the thermal effect are produced due to an increase in the core temperature in the tissue during cell phone exposure. When we are exposed to cell phone, our core temperature of the body and the head is increased, and this depend upon two factors: specific absorption rate and power density. Radio frequency radiations may result in an imbalance between the heat generation and heat dissipation, resulting in thermal effects which could adversely affect the functioning of biological systems. Children are more susceptible to these radiations as they absorb more energy than the adults from the same phone because of their smaller head and brain size, thinner cranial bones and skin, thinner and more elastic ears, lower blood cell volume, as well as greater conductivity of the nerves, due to which the energy penetrate more deeply. You can see here this is a 5-year-old kid, 10-year-old children, and this is an adult. This is the head of 5-year-old children. It shows a deeper penetration of the mobile phone radiation into the head. 10-year-old, little bit less penetration, and adult have a very less as compared to the 5-year-old. So means the childrens, they are very much prone to these radiations. Moreover, the children cell replicate very quickly than adults, which make oncogenes more active. The absorption of these radiations into the head and different parts of the body depend upon the multi-factor like frequency and intensity of the transmission, the duration of exposure, the number of exposures, the distance from the radiation source, the shape and size of the exposed organisms, the water and mineral content of the organism. So let us have a watch over the variables which are used in the measurement of radio frequency radiations. Number one is the power density. It is defined as the amount of power per unit area in a radiated field and is usually expressed in watt per meter square. It is measured by means of a densitometer, radio frequency meter. Radio frequency meter can detect the frequency around us, because the radio frequency now just because of mobile phone, it is also just because of so many other electronic gadgets, lightning in the home, and other equipments which we are used in the home. Second is the SAR value. The term is used to describe the absorption of radio frequency radiations in the body. It is defined as rate of energy that is actually absorbed by the unit of tissue and is measured in watt per kilogram of the tissue. Every mobile phone has a SR rating which has a limit of 1.6 watt per kilogram and is calculated to be 6 minutes per day per person. Following the safety standards for SR limits, a person should not use mobile phone for more than 24 minutes per day. Uh, you can see the maximum limit for mobile phone use is a 24 minutes per day. Unfortunately, this limit is exceeded by information. Our Indian Telecommunication Department has set safety limits of exposure to radio frequency energy produced by mobile phone for general public is 1.6 watt per kilogram. Means we can use maximum of 24 minutes mobile per day. Non-thermal effects continue. The various mechanisms by which the non-thermal effects exert a damaging effect on the tissue are by structural changes of plasma membrane, production of free radicals, changes in ionic transport, and increased DNA damage, and so many other mechanisms. These are shown in the image. So this is the flow chart showing the proposed mechanism of effect of these ionizing and non-ionizing radiations. You can see this thermal effect we have discussed, and non-thermal effect. These radiations affect the living organism by various mechanisms like genetic defects, cell signaling pathway disturb हो सकता है, alteration in the structure and function of cell membrane, increase in the cell defense mechanism, decrease in the cell defense mechanism, increase in the MDSOD and mitotic activated protein kinase, इंट्रासेलुलर सिग्नलिंग पाथवे डिस्टर्ब हो सकता है, एक्टिव एक्टिवेट एपोप्टोटिक पाथवे एक्टिवेट हो सकता है इससे, माइटोकॉन्ड्रिया में इफेक्ट हो सकता है, डीएनए डैमेज है, हीट शॉक प्रोटीन्स, मतलब अल्टीमेटली ऑल दीज थिंग्स दे lead to the structural and functional alteration in the cell, and it is it has been found that these things are uh, directly proportional to the uses. Moreover, some alteration in the body tissue have been recovered if we uh, start keeping ourselves away from these radiations. Harmful of effect of mobile phone radiations. You see here, it may have a short-term problems and a long-term problem. If we talk about the short-term problems, these include headaches, sleep disorders, poor memory, mental excitation, confusion, anxiety, depression, appetite disturbance, and listlessness. Long-term uses of mobile phone includes irreversible hearing loss, damage of the embryonic development, increasing risk of miscarriages, decrease in the number of sperms, damaging of the brain tissues, heart-related problems, weakening of memory, lymphoma, damaging of the genetic structure, drying up of the fluid around the eyes, brain, joints, heart, abdomen, etc., damage to the lens and uveal melanoma, and so many other problems are there because of these radiations. Mobile phone radiation affect the brain function and cause several neurological disorders because the brain is the main target of this mobile phone. Generally, brain, eyes, ear, and reproductive organs, they are more exposed to these radiations. National Institute of Technology, Calicut, Kerala has reported headaches, dizziness, numbness in the thighs, and heaviness in the chest among mobile phone users. PGI Chandigarh has conducted a study and reported long-term intensive mobile phone use may cause internal ear damage. An exposure to radio frequency radiation has been reported to affect physiological, neurological, cognitive, behavioral changes and induce, initiate, and promote carcinogenesis, threat to human health. and has been suggested for mobile phone users. So mobile phone effect on a reproductive system. You see here mobile phone radiation leads to Leydig cell hypoplasia, decrease in the level of serum testosterone, complete absence of spermatozoa, reduced number of ovarian follicles, embryo mortality, oocyte, and DNA damage, which has also been shown by the image. Mobile phone effect on the ovary, endometrium, embryo, then semen quality is also decreased, sperm DNA damage, reactive oxygen species formation, apoptosis rate increase हो जाता है, free radical synthesis also increase, lipid peroxidation is also increased, Leydig cell decreased. So these, as the mobile phones, they are kept in pant pockets very close to the testis and ovaries, reproductive organs. So there is a double effect of these radiations. Whenever we use the phone, our brain is also affected, through which different hormonal level are disturbed. Secondly, mobile phone radiation, they also directly affect the reproductive organs. So in nutshell, we can say that electromagnetic radiations play a very crucial role in our life. Any natural phenomenon is not possible without these radiations. The exposure to cell phone radiation should be reduced as minimum as possible by following accurate safety guidelines, as these may cause irreparable loss to the body. Cell phone radiations are not completely safe. We are always at a risk of greater exposure to non-ionizing radiations while using cell phone. So continuous exposure to these radiation lead to a lot of health ailments, including cancer. So these are some safety tips that can be followed to prevent the exposure to these radiations. Use the handset or speaker. Text more and call less. Call when the signal is strong. Don't put phone in your pocket or other soft area of the body. So thank you very much. Be happy and be radiation free. Thank you.
Thank you. Thank you so much, sir, for sharing such a valuable knowledge regarding radiations of electronic gadgets. And it will be a great pleasure for us to listen you again in our future events. Thank you, sir. Now we have
Thank you
some
very much.
now we have some questions from the participant sides. So I will take each and every question one by one. So the very first question is how 5G radiations and 5G phones affect the human health and up to what extent?
Actually, as I told earlier, that the new latest generation mobile phone, they have a main focus on the speed and performance. The 5G mobile phone, they are equipped with multiple antennas to boost the speed and performance of the mobile phone. So definitely, they will uh, we will be exposed more to these radiations while in a era of 5G revolution. So definitely, they will harm. The different research are going on.
Thank you, sir. One more question is there. What are some very useful tips for avoiding the usage of phones and electronic gadgets?
Mobile phone, these are, you can say, fourth basic need of life. We can't avoid mobile phone, but we have to be very smart. The phone becomes smart, but we become dull. So we have to be very smart. We have to use the phone smartly. We have to reduce the usage of phone to minimum, and use Bluetooth, uh, avoid unnecessary uh, viewing the entertainment media, etc. Only use the phone for its basic applications and essential applications. Do not attend the call in a poor network. When there is a poor network, the phone try to contact with the different antennas. At that time, the phone emit more and more radiations. And some sort of screen savers are also advised by different uh, companies, and that is not sure whether they screen these radiations or not. So this is a very basic thing that everybody should know, and everybody have the idea about. But these are actually, problem is that these are hidden things, and we are always surrounded by these things, and we are not curious about these things, and we should take necessary precautions.
Okay, thank you, sir. Thank you for answering in such a nice way. Now, rest of the question we will discuss in a WhatsApp group of that symposium. Now we have our second speaker with us, the very eminent speaker, Dr. Vijit Agarwal.
Thank you
Sir
very
is,
much.
sir is currently working as a senior pharmacovigilance associate and team leader at National Coordination Center, Pharmacovigilance Program of India, Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, Government of India, Ghaziabad, Uttar Pradesh. Sir has more than 6 years of experience in research, pharmacovigilance, and teaching sector. Sir has also published a number of paper and abstract in national journal and also attended many national and international conferences. Now, may I request Dr. Vijit Agarwal to please address the esteemed participants with his valuable knowledge on the topic career opportunities in pharmacovigilance. Over to you, sir.
Thank you. Thank you, Miss Aman. Hello, everyone. Good morning. I would like to thank you for giving me this opportunity to be on this platform. I would thank all the organizers, the Vice Chancellor, Chancellor, Pro Chancellor, each and everyone who have made this program a successful one. And I hope all the participants whosoever are joining this session would surely be benefited. Uh, so today our topic of discussion would be career opportunities in pharmacovigilance. So it is like one of the very important segment these days. So let me share with you my screen of PowerPoint presentation. Just give me a second. So this my screen is visible, and am I audible to you?
Yes, sir, you are audible as well. Screen is also visible.
So thank you, Aman. So today our this topic of discussion would be career opportunities in pharmacovigilance. It is one of the most important part of all the life cycle of the drug. So in this symposium, all of you must have got an idea what is the process of drug development. So the drug starts with the preclinical studies. That is, before that also a lot of things happens. But when we talk about the clinical and preclinical studies, we start with the preclinical one. So where the animals and the drug will be tested on the animals, and later on it will be tested on human beings. After that, um, after that drug is sent for the approval to the regulatory authority. So in case of our country in India, the regulatory authority is Central Drug Standard Control Organization. So it's the head office in is in New Delhi. So there uh, the application will be sent for the approval. Once the drug is approved for marketing, so later on it will be available throughout the country, in all over, from where the public can have the access for these drugs. So today, first of all, about pharmacovigilance, what is it? It is also called as phase 4 clinical studies, you can say, or post marketing surveillance and pharmacovigilance. These all are synonymous terms. And the WHO, the World Health Organization, has also defined pharmacovigilance as the science and activities relating to the detection, where you are going to detect the problem, and assessment. You have to assess the problem, then understand it, and then you have to come up with a prevention plan, how the drug, or how the adverse effect which has been happened due to the drug, and or any other kind of drug related problem can be resolved. So तो सिंपल लैंग्वेज में आई वांट टू टेल कि सबसे पहले हमें इसे डिटेक्ट करना है, समझना है कि मतलब प्रॉब्लम क्या है, कहाँ पे प्रॉब्लम हुई है, उसको डिटेक्ट करना है। लेटर ऑन असेस करना है कि कई बार जब हम क्लिनिकल ट्रायल्स की बात करते हैं, तो क्लिनिकल ट्रायल्स में एक बहुत ही कंट्रोल्ड एनवायरनमेंट होता है। उस कंट्रोल्ड एनवायरनमेंट में हमारे को पता नहीं लगता कि एक्चुअल रियल वर्ल्ड सिचुएशंस में क्या प्रॉब्लम आएगी, कहाँ प्रॉब्लम आएगी, और उसके बाद असेस करना है कि वाकई में इस प्रॉब्लम की वजह ये ड्रग ही है या कुछ और प्रॉब्लम हो सकती है, कुछ और वजह से ये प्रॉब्लम हुई है। Then the whole scenario should be understood by the pharmacovigilance expert, or the clinician, whosoever is treating the patient, or the consumer. And then it comes to the prevention. Then prevention means अगर ये पर्टिकुलर एडवर्स इवेंट रोका जा सकता है, तो उसके लिए हमें प्रिवेंशन प्लान्स बनाने पड़ेंगे। So the whole science of this activity is called as pharmacovigilance. So I hope कि क्योंकि करियर समझने से पहले हमें समझना पड़ेगा पीवी क्या है, उसको तभी हम आगे करियर के बारे में हम प्लान कर सकते हैं। So the prevention of adverse effects or any other drug related problem, uh, all comes under this pharmacovigilance. Later on, in simple terms, you can say pharmacovigilance involves a number of activities which are aimed at identifying the risks associated with the medical products. So the medical products can be anything like drugs, vaccines, or medical devices, and ensures that their harmful effect on the patients are prevented or minimized. हम या तो उसे कम कर दें या तो रोक दें। रोकना तो बहुत अच्छी बात है, कम भी कई केसेस में ऐसा होता है, जैसे अभी मैं आपको एक एग्जांपल देता हूँ जिसमें कि आप उसको मिनिमाइज करने की बात कहते हैं। जैसे कि कई बार बात करते हैं एंटी कैंसर ड्रग्स की, तो उसमें पेशेंट्स बहुत ज़्यादा ऐसी ड्रग्स ले रहे होते हैं कि उनको बहुत सारे जीआई साइड इफेक्ट्स,
Sir, sorry to interrupt. Sir, sorry to interrupt, but your slides are not showing. Only the in screen, only the first slide is visible.
This where the heading was mentioned.
Yes, sir. The career opportunity.
Is this moving now?
No, sir.
No. Not moving.
No, sir. Only first slide is.
Okay, wait a minute.
Yes, sir. It's moving now.
Is it correct now? Is it visible?
Yes, sir.
Moving?
Yes, sir.
Okay. So, in the simple terms, we can say pharmacovigilance involves a number of activities aimed at identifying the risks. हमें risk को identify करना है, क्या risk हो सकते हैं associated with the medical products, जो कि medical products से जुड़े हुए हैं, और उनको इंश्योर करना है कि उनका जो हार्मफुल इफेक्ट है वो पेशेंट्स पे या तो कम से कम पड़े या बिल्कुल हम प्रिवेंट कर सकें। तो इसका एग्जांपल मैंने आपको बताया कि एंटी कैंसर ड्रग्स के अंदर कई बार ऐसे जीआई सिम्टम्स होते हैं, और उसके आपने देखा होगा कई बार यूजुअली एंटी कैंसर ड्रग्स के ट्रीटमेंट में एलोपेशिया बहुत कॉमन है। तो उस केस में क्या होता है, जो कैंसर से पेशेंट जूझ रहा होता है, उस केस में पहले तो वो कैंसर की मार सह रहा होता है, साथ के साथ अगर ऐसी मेडिसिन चलेंगी तो उससे और ज्यादा साइड इफेक्ट्स, प्लस उसके अलावा सोशल बहुत ज्यादा कट ऑफ होता है, उसके लिए उसका साइकोलॉजिकल कंडीशंस के ऊपर बहुत असर पड़ता है। तो इन सब चीजों को हमें प्रिवेंट करना है। किस तरीके से हम कर सकते हैं कि अगर हमें पता हुआ कि हम हमारे जैसे गैस्ट्रो इंटेस्टिनल प्रॉब्लम्स आने वाली है, या कुछ गैस्ट्रिक डिस्कम्फर्ट इट इज गोइंग टू कॉज, सो वी स्टार्ट विद some kind of drugs which resolves those problems. So हम उसको या तो डोज मिनिमाइज करेंगे, या डोज को चेंज करेंगे, डोज को अल्टर करेंगे, या कुछ ऐसी ड्रग्स देंगे साथ में जो हमारे गैस्ट्रिक डिस्कम्फर्ट को रिलीफ देंगी। तो ये सब प्लान्स जब निकल के आते हैं, जब हमारी पोस्ट मार्केटिंग सर्वेलेंस में स्टडीज जब हो रही होती है, तब हमें पता लगता है कि हाँ, कि हम इस तरीके से भी पेशेंट को मतलब हार्म को मिनिमाइज कर सकते हैं। प्लस हमारे को ये भी ध्यान में रखना होता है कि पेशेंट के ऊपर जो हमारा अ, अ, जो ड्रग्स का जो बर्डन है वो कम से कम रहे, पॉली फार्मेसी ना हो, ज्यादा बहुत ज्यादा ना हो, क्योंकि जब रियल वर्ल्ड सिनेरियो में आते हैं तो पेशेंट जरूरी नहीं है केवल उसी ड्रग के प्रॉब्लम। जैसे फॉर एग्जांपल मैं आपको देता हूँ, जब एंटी हाइपरटेंसिव ड्रग्स का ट्रायल हो रहा होता है तो उस दौरान पेशेंट्स केवल और केवल हाइपरटेंसिव पेशेंट्स को ही इंक्लूड करते हैं स्टडी के अंदर। बट व्हेन इट कम्स टू द रियल वर्ल्ड सिनेरियो, तो मोस्ट ऑफ द केसेस में आपको 2 से 3 प्रॉब्लम्स उनमें मिलेंगे ही मिलेंगी। तो उन सिचुएशंस में ट्रायल का डेटा हमारे पास अवेलेबल नहीं होता है, तो वी कैन नॉट डू ट्रायल। तो ट्रायल क्या है, बहुत ही साफ सुथरा सा रोड है कि हमें सीधा सीधा जाना है। बट व्हेन इट कम्स टू द रियल वर्ल्ड सिनेरियो, वो बहुत ही क्राउडेड मामला हो जाता है। तो उस केस में हमें बहुत ध्यान रखने की जरूरत है, और पेशेंट को मिनिमम ड्रग्स के साथ, मिनिमम साइड इफेक्ट के साथ ट्रीट भी करने का एम होता है, जो सब कुछ हमारा फार्माकोविजिलेंस में आता है। तो एक बहुत बड़ा साइंस है देखा जाए तो, इसको हम एक दिन में नहीं सीख सकते, बट हाँ, प्रोसेस के साथ इसको सीखा जा सकता है। तो हमारे को फार्माकोविजिलेंस क्यों चाहिए, व्हाई डू वी नीड इट? सो first of all, the increased number of drugs and biologics entering into the market. तो अभी आपने देखा होगा कोविड के टाइम में कितने सारे ड्रग्स को मार्केट के अंदर लाया गया, और मार्केट में वो ड्रग्स मार्केट भी हुई हैं, और कई ड्रग्स को इमरजेंसी यूज ऑथराइजेशन भी दिया गया। तो ये सब के आने की वजह से हमको जो रेगुलेटरी अथॉरिटीज होती हैं, उनको चाहिए होता है उनका डेटा, कि वाकई में रियल वर्ल्ड सिनेरियो में क्या पिक्चर है इसकी पर्टिकुलर मेडिकल प्रोडक्ट की। तो उस प्रोडक्ट के बारे में उसको जो रेगुलेटरी अथॉरिटी होती है उस कंट्री की, या जिस भी कंट्री में वो ड्रग यूज हो रही है, उनको उसका डेटा चाहिए होता है। तो ये फार्माकोविजिलेंस की वजह से ही मिलता है हमको। दूसरा है enhanced drug safety regulatory framework, so which I have just explained. Now the third one is increased drug safety awareness. तो अभी आज की डेट में क्या है, बहुत अवेयरनेस भी बढ़ रही है लोगों में। तो अभी डॉक्टर के पास जाने से पहले भी कई पेशेंट्स गूगल करके जाते हैं। तो आपने देखा होगा कई बार दे स्टार्ट आर्ग्यूइंग विद द डॉक्टर्स समटाइम्स। तो इन दो सिनेरियोस अगर आप देखेंगे तो होल सोल अगर आप देखेंगे तो ड्रग सेफ्टी के लिए अवेयरनेस बढ़ रही है। तो हमें ये चीज का हमें इस चीज की वजह से भी फार्माकोविजिलेंस का डेटा चाहिए। क्यों चाहिए? कि मान लीजिए कोई क्लेम करता है कि नहीं, हमारे को तो लगता है कि ये ड्रग ये भी कॉज करेगी, ये भी कॉज करेगी, तो हम वी हैव सम डेटा, द फार्माकोविजिलेंस डेटा टू शो टू द पेशेंट कि नहीं, आपका ये जो आप देख रहे हो ऐसे तो नहीं है। इस तरीके से हमारे पास डेटा भी है, वी हैव दैट डेटा विच defies that what you are claiming is wrong, this is the right thing which we have. So these are the things why we need pharmacovigilance. So what are the basic requirements to be a pharmacovigilance professionals? There are two things. First, the technical skills. Next is the soft skills. So when we talk about the technical skills, so it is not only limited to the degree. However, degree is a necessary requirement, like degree in pharmacy, degree in life sciences, degree in medicine and nursing. But instead of that, some technical skills are mandatory. Like most of the times, when we talk about PV professionals, anyone can enter into the pharmacovigilance field. Any person, if when we talk about only pharmacy, I'm not going to talk about life sciences, medicine, nursing, but I'm talking about the pharmacy profession, where many times M.Pharm in chemistry, M.Pharm in zootics, people also enter into the pharmacovigilance profession. But after gaining a good amount of experience, they become really what to say, really expert in the area of pharmacovigilance. And the next but not the least is soft skills. In the soft skills, you must have the confidence, a team player. The person must be a team player. The PV professional must have work ethics, and he or she should be a problem solver. So why am I talking about the soft skills? Is because pharmacovigilance is an industry based part. It is not some kind of what to say, any simple kind of job. जब हम बात करते हैं सॉफ्ट स्किल्स की, तो जब हम बात करते हैं, तो हम बात करते हैं इंडस्ट्री के अंदर हम किस तरीके से हमारे पास सॉफ्ट स्किल्स होने चाहिए, हमारे को तभी हम आगे ग्रो कर सकते हैं. Otherwise, only mere having a degree in pharmacy or life sciences will not make you excel in pharmacovigilance profession. So the PV professionals के कई सारे functions हैं. So a typical pharmacovigilance operation will have various functions like individual case safety report processing. So the very first part which comes for in the pharmacovigilance field is individual case safety report processor. So here, what is this ICSR? ICSR is a case where some प्रॉब्लम हुई और किसने की है, ये सब हम बताते हैं. तो आईसीएसआर में भी वही है. तो आईसीएसआर में क्या करना है, प्रॉब्लम क्या हुई है, कल्प्रिट क्या है, किस सिचुएशन में हुआ, क्या हमने डोज लिया था, क्या एक्चुअली हुआ था, किस समय पे हुआ था, कब हमने ड्रग लिया था. तो ये सब सिचुएशंस हमें बताना होता है आईसीएसआर के अंदर. तो ये जब डेटा हमारे पास फॉर्म के अंदर आता है, तो ये सबसे पहला पार्ट होता है फार्माकोविजिलेंस फंक्शन का. लेटर ऑन जब हमारा आईसीएसआर हमारा प्रोसेस हो जाता है, इसको एक क्वालिटी चेक जो है क्वालिटी चेक करता है, उसके वो भी प्रोसेसर ही कहलाता है. तो क्वालिटी चेक जब किया जाता है, तो उसमें ये देखा जाता है कि सारे फील्ड्स, एक्चुअली जो आईसीएसआर है वो ऐसा नहीं है कि किसी सिंपल एक्सेल शीट में हमने भर दिया. इसके लिए डिफरेंट सॉफ्टवेयर होते हैं, लाइक आर्गस है, इरिजी है, इन WHO इट्स विजीफ्लो. तो ये डिफरेंट सॉफ्टवेयर्स हैं. तो यूजुअली इंडस्ट्रीज के अंदर आर्गस और एरिजी का यूज किया जाता है. तो आर्गस और एरिजी में जब ये डेटा एंटर किया जाता है, उसके बाद एक क्वालिटी चेक किया जाता है. तो जो भी आईसीएसआर प्रोसेसर्स को जिनको एक्सपीरियंस हो जाता है, लेटर ऑन दे बिकम क्वालिटी चेक क्वालिटी रिव्यूअर्स. तो जब वो क्वालिटी रिव्यू करते हैं केस का, तो उसमें कुछ भी प्रॉब्लम्स जो भी फाइंड करते हैं या कुछ भी मिसिंग फील्ड्स हैं, तो उनको करेक्ट करते हैं. लेटर ऑन इट कम्स फॉर द मेडिकल रिव्यू. तो उसका मेडिकल रिव्यू किया जाता है केस का. ये भी एक फार्माकोविजिलेंस का फंक्शन है. लेटर ऑन एक और चीज होती है जो हमारे केस को निकालने के लिए, एक तो होती है पेशेंट ने बता दिया. कई बार क्या होता है, जो लिटरेचर में भी हमारी कई एडीआर्स को रिपोर्ट किया जाता है. लिटरेचर यानी कि हमारे पब्लिकेशंस के अंदर. तो उस पब्लिक तो इंडस्ट्रीज का एक और काम होता है कि वो लिटरेचर को सर्च करते हैं, उसका रिव्यू करते हैं, और जितने भी उनके प्रोडक्ट रिलेटेड एडवर्स रिएक्शंस आए हुए होते हैं या उन्हें सर्च करने पर मिलते हैं, उनको भी रिपोर्ट करने का काम भी इंडस्ट्रीज का होता है. तो यहाँ पर भी हमारा फार्माकोविजिलेंस फंक्शन एक आता है. लेटर ऑन एग्रीगेट रिपोर्ट प्रिपरेशन. तो एग्रीगेट रिपोर्ट्स क्या होती हैं, पेब्रर हो गया, पीएसयू पीरियोडिक सेफ्टी अपडेट रिपोर्ट्स हो गया, या डेवलपमेंट सेफ्टी अपडेट रिपोर्ट्स हो गया. तो ये सब पीएसयूआर और डीएसयूआर का सबमिशन भी एग्रीगेट रिपोर्ट प्रिपरेशन करने का काम भी फार्माकोविजिलेंस प्रोफेशनल्स का होता है. तो तो ये सब वेज हैं कि कहाँ की रिपोर्ट हमारे पास आ सकती है, और फिर हमें वो जब हमारे पास रिपोर्ट आ जाएंगी तो हमें उसका क्या करना है, यानी कि एग्रीगेट करना है उन्हें। मतलब एग्रीगेट रिपोर्ट उतना बड़ा जैसे देखने में लग रहा है उतना बड़ा है नहीं, सिंपल समझने वाली चीज है। हमें एग्रीगेट करना है, मतलब समझना है कि हमारे पास मान लीजिए 20-25 केस हमें मान लो एक 3 महीने में या 6 महीने में मिले उस पर्टिकुलर ड्रग के एडीआर के रिलेटेड। अब हमें उसको एक समराइज वे में बताना है, पीरियोडिक सेफ्टी अपडेट रिपोर्ट्स। तो हमें सेफ्टी अपडेट करनी है पीरियोडिकली। तो वो पीएसयूआर का मतलब होता है कि उसको हमें एक समराइज्ड रिपोर्ट बनाना है जिसका नाम है पीएसयूआर, तो ये पीरियोडिक अपडेट सेफ्टी अपडेट रिपोर्ट्स। तो इसमें जब कोई केमिकल एंटिटी नई मार्केट में एंटर करती है, तो जो करंट इंडियन गाइडलाइंस कहती हैं, वो इंडियन गाइडलाइंस ये कहती हैं कि जो भी न्यू एंटिटी मार्केट में आएगी तो उसका जो पीएसयूआर सबमिट करना है उसको पहले 2 साल तक एवरी 6 मंथ्स करना है, लेटर ऑन एवरी ईयर फॉर 2 years करना है। उसके बाद अगर रेगुलेटरी अथॉरिटी कहती है तो सबमिट करना, अदरवाइज देर इज नो नीड टू सबमिट दैट पीएसयूआर और डीएसयूआर। सो और एज पर द रिक्वायरमेंट, जो भी रिक्वायरमेंट कभी कभी रेगुलेटरी अथॉरिटी बोल देती है कि हमें ये ड्रग का आपको ये चाहिए पीएसयूआर इसका, तो वो भी सबमिट करना पड़ता है। लेटर ऑन कम्स टू द सिग्नल डिटेक्शन, फार्माको एपिडेमियोलॉजी एंड रिस्क मैनेजमेंट। तो ये सब जो अपने आप में सेल्फ डिफाइंड ये अपने आप में है कि सिग्नल डिटेक्ट करने के लिए आपको क्या करना होगा। इसमें कई तरह की स्टैटिस्टिकल एनालिसिस लगता है। उसके बाद आप एक कंक्लूजन में पहुंचते हो कि इस पर्टिकुलर ड्रग का सिग्नल कोई पर्टिकुलर एडवर्स इफेक्ट जो कि क्लिनिकल ट्रायल्स के दौरान नहीं मिला था, वो कुछ नया आपको मिला है, तो वो सिग्नल डिटेक्शन में आता है। और फार्माको एपिडेमियोलॉजिकल स्टडीज मतलब आपको पब्लिक के ऊपर जो एपिडेमियोलॉजिकल स्टडीज करनी है वो आती हैं, प्लस रिस्क मैनेजमेंट आपका, जो भी आपको कोई रिस्क मैनेजमेंट प्लान के साथ आना है कि कोई पर्टिकुलर ड्रग कोई ऐसा एडवर्स रिएक्शन कॉज कर रही है बट उस ड्रग का यूज भी बहुत जरूरी है, उस केस में रिस्क मैनेजमेंट प्लान भी होने चाहिए हमारे पास। तो वो कंपनीज का ही काम होता है, मैन्युफैक्चरिंग फर्म्स का ही काम होता है जो उसे सबमिट करें। देन कम्स टू द लेबलिंग एंड क्वालिफाइड पर्सनल फॉर फार्माकोविजिलेंस। सो अभी मैं बताऊंगा इसको थोड़ा सा और डिटेल में। तो जैसा आपको आईसीएसआर प्रोसेसिंग का मैंने आपको बता ही दिया, लाइक फर्स्ट ऑफ ऑल कलेक्शन एंड ट्रायज ऑफ एडवर्स रिएक्शन इन द सेफ्टी डेटाबेस। Then we have to perform some duplicate checks and data entry must be done. Then the quality check should be done. Then data coding. So in the coding part, adverse events and drug coding must be there. So for adverse event coding, there is a dictionary called MEDRA. So that MEDRA must be used. So all these softwares have that subscription of MEDRA. So whatsoever adverse event is um, reported, that must be coded in terms of verbatim. Means whatever the patient or the consumer or the physician have reported it, that must be coded. Now comes to the drug coding part. Drug also must be coded. For this, a dictionary is used called WHO Drug Dictionary, so that drug should be coded. So without coding, there is no use of that data. So this is whenever you goes into this profession of pharmacovigilance, you will come to know what how the adverse event should be coded, how the drugs are coded, what is the causality assessment, and how the follow up happens. Then data reconciliation and expedited reporting also comes into the ICSR processing part. Next, come to the literature search and review. So here, all the what to say, literature will be searched by the manufacturing firm or वो जो भी है उसका थर्ड पार्टी जो भी उसने हायर कर रखी है फार्मास्यूटिकल फर्म ने, तो वो उनका लिटरेचर सर्च का काम करेंगे और रिपोर्ट करेंगे। नाउ मेडिकल रिव्यू आता है। मेडिकल रिव्यू में आपका जो भी पीरियोडिक रिपोर्ट्स होती हैं या फिर आईसीएसआर होते हैं उनका मेडिकल रिव्यू करना होता है, और उसके उसमें करते हैं। Now comes to the aggregate report preparation. तो इसमें आपको पेब्रर या फिर डेवलपमेंट DSUR, पीरियोडिक बेनिफिट रिस्क असेसमेंट रिपोर्ट्स और डेवलपमेंट सेफ्टी अपडेट रिपोर्ट्स और जो भी आपको रिपोर्ट्स हैं वो एग्रीगेट रिपोर्टिंग में आता है। तो उसको जब मैंने अभी बताया था आपको जो समरी होती है उसको समरी को एक तरह से बनाना होता है। इसका भी एक डेटा फॉर्मेट होता है पर्टिकुलर जो कि रेगुलेटरी अथॉरिटी बताती है कि इस तरीके से बनाना है, हमको इस तरीके की रिक्वायरमेंट है। तो फार्मास्यूटिकल फर्म का काम होता है उस तरीके से बना करके देना। तो यहाँ पर आपकी जॉब अपॉर्चुनिटीज इस तरीके से क्रिएट होती है। फार्मास्यूटिकल फर्म्स PV प्रोफेशनल्स को हायर करती है, फार्मा फार्मा प्रोफेशनल्स को हायर करती हैं ये सब काम करने के लिए। बट ये एक दिन का टास्क नहीं होता। आदमी मतलब हमें इसमें थोड़ा टाइम लगता है सीखने के लिए। But when you are into this profession, ICSR processing and literature search or medical review, when you are efficient with all of these, you will also be a part of aggregate reporting, where you will learn about PEBRAR or DSUR, how to make all of these reports. Now then comes to the signal detection, pharmacoepidemiology, and risk management. This is also I have already touched upon, where the screening of various data sources such as clinical trials and spontaneous reporting system, literature, external databases, and others to identify the signals through the data mining. So data mining is also one process where you will come to know how data should be mined, and after the data mining, how the person, how the pharma or PV professional will come up with a signal detection. And then after detection of the signal, we have to validate the signal, then prioritize it and evaluate it. The after all of this preparation of risk management plans or risk evaluation and mitigation strategies जो कि बनानी होती है हमें, तो वो भी इसी में आती है, इसी का पार्ट होता है। और जब हम ये सब कर लेते हैं तो हमें फिर उसको कम्युनिकेट भी करना होता है। कैसे कम्युनिकेट होता है उसको, जैसे मान लीजिए कोई एक ड्रग के साथ हमें कुछ नया साइड इफेक्ट उसका मिला, तो उसको हम बाद में रेगुलेटरी अथॉरिटी को कम्युनिकेट करते हैं। तो लेटर ऑन रेगुलेटरी अथॉरिटी उस फार्मास्यूटिकल फर्म को कम्युनिकेट करती है कि आपको अपने PIL में इस तरीके से अपडेट करना है। तो वो चीज बहुत एक लेंदी प्रोसेस होता है, बट ये बहुत मस्ट प्रोसेस है, करना पड़ता है सारे फार्मास्यूटिकल फर्म्स को, और ये इस तरीके से बाद में उसको अपडेट करा जाता है। कई केसेस में क्या होता है, कोई बहुत ही सीरियस कंडीशन है, बहुत सीरियस एडवर्स इवेंट कॉज हो रहा है, तो उसमें उस केस में ड्रग्स को मार्केट से उठाना भी पड़ता है, हटाना भी पड़ता है। वो एक बहुत एक्सट्रीम कंडीशन होती है जो कि बहुत रेयर कंडीशंस में आती है, जैसे आपने थैलिडोमाइड के बारे में शायद सुना होगा। तो नाउ कम्स टू द लेबलिंग। इन द लेबलिंग जो मैंने अभी आपको बताया कि उसमें आपको क्रिएट करने पड़ेंगे, मेंटेन करने पड़ेंगे कंपनी प्रोडक्ट्स के लेबल्स, कंपनी कोड, डेटा शीट्स एंड ऑल। उसके बाद आप उसके पैकेज इंसर्ट्स को, पेशेंट इंफॉर्मेशन लेफलेट्स को आप उसको अपडेट करेंगे। तो ये सब काम भी फार्माकोविजिलेंस प्रोफेशनल्स का होता है। हाँ, ये काम सबका बहुत ही डिवाइडेड तरीके से होता है। ऐसा नहीं है कि जो प्रोसेसिंग कर रहा है वही सिग्नल डिटेक्शन कर रहा है, वही लिटरेचर को सर्च कर रहा है, ऐसा नहीं है। हर एक टीम का काम बटा हुआ होता है, और टीम वाइज जिस फील्ड में भी आपको ये लगता है कि हाँ ये मेरे टाइप का फील्ड है, तो आप उसमें धीरे धीरे करके एंटर होते रहते हैं। नाउ कम्स टू द QPPV। तो अभी आपको पता होगा कि 1 सेकंड प्लीज। हेलो, हाँ जी, बाद में याद करता हूँ। Are my slides visible to all of you?
Sir, now it is visible. 8th slide. Number 8 is visible.
Yes. So where the labeling and QPPV is mentioned, right?
Yes, sir.
So now when we talk about the QPPV, it is qualified person for pharmacovigilance. So this is one of the most important post. तो अभी यहाँ पे मैं आपको बताऊंगा दो चीजें होती हैं, दो तरीके से फार्माकोविजिलेंस इंडस्ट्री काम करती है, जो सबसे इम्पोर्टेंट है। फार्माकोविजिलेंस इंडस्ट्री खुद ही फार्माकोविजिलेंस का मतलब फार्मास्यूटिकल फर्म्स जो होते हैं, या तो वो खुद ही अपना फार्माको विजिलेंस एक्सपर्ट को अपने यहाँ पे हायर करते हैं, पूरी टीम बनाते हैं और उससे काम कराते हैं, या फिर फार्मास्यूटिकल फर्म अपने प्रोडक्ट की विजिलेंस के लिए किसी थर्ड पार्टी को हायर करती है। वो थर्ड पार्टी का काम होता है उसके ऊपर वॉच रखना और उस कंपनी को, उस फार्मास्यूटिकल फर्म को रिपोर्ट सबमिट करना। तो ये दो तरीके से होता है। तो या तो हम उसको खुद करें, या हमने उस पर्टिकुलर प्रोसेस को थर्ड पार्टी कर देते हैं। तो दोनों चीजों में अलग अलग काम होता है। थर्ड पार्टी का अलग काम होता है, थर्ड पार्टी अलग तरीके से काम करते हैं, जैसे कि सीआरओज हो गए, जो कॉन्ट्रैक्ट रिसर्च ऑर्गेनाइजेशन होते हैं, वो सब अपना काम अलग तरीके से करते हैं। वो बहुत ही इंडस्ट्री बेस्ड काम होता है। और एक जो होता है फार्मास्यूटिकल फर्म्स खुद करते हैं, वो अपना क्यूपीपीवी रखते हैं और वो अपना कंप्लीट टीम बनाते हैं। तो तो इसमें मैं क्यूपीपीवी के बारे में बता रहा हूँ, जिसमें कि आपका ओवरसाइट ऑफ द कंपनी फार्माकोविजिलेंस सिस्टम एंड प्रोसेसेस, and then overview of the safety profile of the product and communicate with the health authorities as required. तो वो पॉइंट ऑफ कांटेक्ट होता है हेल्थ अथॉरिटीज के लिए। जब भी कोई फार्मास्यूटिकल फार्माकोविजिलेंस रिलेटेड एक्टिविटी होगी, या कोई भी फार्माकोविजिलेंस रिलेटेड डेटा रेगुलेटरी अथॉरिटी को चाहिए होगा या हेल्थ अथॉरिटी को चाहिए होगा, वो सिंगल पॉइंट ऑफ कांटेक्ट यूपीपीवी को आपके फार्मास्यूटिकल फर्म में कांटेक्ट करेंगे डायरेक्टली। तो ये काम होता है। और ये आपने अभी रिसेंटली, अगर आपने सीडीएसओ की वेबसाइट अगर आपने देखा हो, तो सीडीएसओ की वेबसाइट पे करंटली एक ऑर्डर भी आया है, जिसमें वो मेंशन कर रहे हैं कि जितने भी फार्मास्यूटिकल फर्म्स हैं, उनको एक क्यूपीपीवी को अपने यहाँ पर हायरिंग करना ही करना है। तो अभी ये वैक्सीन के लिए आया है, लेटर ऑन हो सकता है फार्मास्यूटिकल फर्म्स के लिए भी आए। हालांकि ये चीज मेंशनड ऑलरेडी है हमारे एनडीसीटी 2009 में, न्यू ड्रग्स एंड क्लिनिकल ट्रायल्स 2019 में जो रिलीज किए थे सीडीएसईओ ने, मिनिस्ट्री ने। तो वो चीज ऑलरेडी मेंशनड है वहां पर कि क्यूपीबीवी फार्मास्यूटिकल फर्म में होना ही चाहिए, जो कि हेल्थ अथॉरिटीज का डायरेक्ट कांटेक्ट होता है। बट अभी तक उसको उतना इफेक्ट में नहीं आया है। कई फार्मास्यूटिकल फर्म्स ने उसको अपने यहाँ पर इम्प्लीमेंट करना शुरू भी कर दिया है, बट अभी भी धीरे धीरे वो चीज प्रोसेस में है। नाउ the pharmacovigilance offers plenty of opportunities for career development in pharmaceutical industry. With increased number of drugs and biologics entering into the market and enhanced drugs safety regulatory framework, it needs for a skilled resources to carry out the pharmacovigilance activities, which has gained a lot of importance. The increased drug safety awareness has also demanded robust and cost-effective pharmacovigilance system and operations. As a result of this, pharmacovigilance outsourcing has gained momentum. Thus, it is increasing the job opportunities. तो अभी जैसे कि मैं आपको इसमें आप देख रहे हो, एक लेटर आपको दिखाई दे रहा होगा। तो हम लोग जब शुरू करते हैं, जो हमारे पास जब हम फ्रेशर होते हैं, तो वी कम अप विद लाइक फार्माकोविजिलेंस और ड्रग सेफ्टी कोऑर्डिनेटर वी स्टार्ट विद 1 to 2 years of experience will be required. Um, later on, we become associates, and then we become PB or pharmacovigilance or drug safety specialist or scientist. So जैसे जैसे हमारे पास एक्सपीरियंस बढ़ता जाएगा, लाइक यू कैन सी इन द 10+ years of experience, the pharmacovigilance expert can become a senior pharmacovigilance drug safety manager. So so this is how the career ladder will work. And अगर आप उसमें कंटिन्यूअसली एक्सेल करते रहते हैं, अपने सॉफ्ट स्किल्स को डेवलप करते रहते हैं, अपने टेक्निकल स्किल्स को इनहांस करते रहते हैं, आपके पास जितने ज़्यादा से ज़्यादा आपके पास लर्निंग्स होंगी, एक्सपीरियंसेस होंगे, तो आप ज़्यादा से ज़्यादा बड़े रोल्स के लिए अप्लाई कर सकते हैं और उसके लिए आप काम कर सकते हैं। तो आप देख सकते हैं कि 10-15 ईयर्स के एक्सपीरियंस के बाद, 20 ईयर्स के एक्सपीरियंस के बाद सीनियर डायरेक्टर और वाइस प्रेसिडेंट पोस्ट मिलता है, जो कि यूजुअली सीआरओ और इंडस्ट्रीज के अंदर हम देखते हैं ये होता है। तो ये होल सोल एक हमारे को एक बताता है कि किस तरीके से फार्माकोविजिलेंस के अंदर आप अपना कैरियर एक्सेल कर सकते हैं। थैंक यू सो मच।
Thank you so much, sir, for sharing such a valuable knowledge in pharmacovigilance. This would definitely help our pharmacy students. And we have some queries from the participant sides. So the very
Yes.
first question is, as a pharmacy career option, which one is best, pharmacovigilance or a regulatory sciences?
सो इसमें आपको बोलूंगा रेगुलेटरी फार्माकोविजिलेंस इज द पार्ट ऑफ रेगुलेटरी सिस्टम. तो रेगुलेटरी क्या है ना, एक बड़ा बड़ा पार्ट है, उसका एक छोटा सा पार्ट है फार्माकोविजिलेंस. तो रेगुलेटरी के अंदर आपको काफी मल्टी डाइवर्स आपको काम करने को देखने को मिलेंगे. उसमें सबमिशन अलग अलग तरह के आते हैं, डोजियर सबमिशन से लेकर के सेफ्टी सबमिशन तक आते हैं, सभी कुछ आता है. बट व्हेन इट कम्स टू द फार्माकोविजिलेंस, तो वो फार्माकोविजिलेंस एक सिंपल एक छोटा सा पार्ट है. हालांकि फार्माकोविजिलेंस अपने आप में बहुत बड़ा है, वास्ट है, बट रेगुलेटरी के लिए एक छोटा पार्ट है कि फार्माकोविजिलेंस के अंदर खाली सेफ्टी के लिए हम डील करेंगे, नॉट मोर देन दैट. हाउएवर फार्माकोविजिलेंस को साथ के साथ काम करना पड़ेगा रेगुलेटरी के और मैनेजिंग टीम्स के और अदर टीम्स के साथ इंडस्ट्री की, बट वो एक स्मॉल पार्ट है, रेगुलेटरी एक बड़ा पार्ट है. तो आई वुड से अगर आप रेगुलेटरी से शुरू करें तो इट वुड बी बेटर. और अगर आप को फार्माकोविजिलेंस में आपको लगता है कि फार्माकोविजिलेंस विल वुड मेक एन इम्पैक्ट, तो ऑब्वियसली व्हेन यू आर इन द रेगुलेटरी यू कैन मूव एनीवेयर, like in pharmacovigilance, or you can be stay in regulatory part itself.
Okay, thank you, sir. The second question we have is, beside the pharma professional, who else will opt this career? Whosoever will opt a pharmacovigilance as a career?
Like as I've already told, pharma people, and if someone have life sciences degrees like BSc, MSc, they can also, because currently uh, many university are delivering this course like MSc in pharmacovigilance, M.Pharm in pharmacovigilance, M.Pharm in drug regulatory authority, regulatory affairs, and medical professionals, MBBS, BDS doctors, and even physiotherapists sometimes. So many, but the person should be associated with some kind of science field. Mathematic field may not be that much okay for this pharmacovigilance function. Some professionals who have some kind of medical knowledge, so those can enter into this field.
Okay, thank you, sir. Sir, one more question is there. Ma'am, please.
So there is a causality is a relationship between the suspect product and the adverse drug event. So can you give an example about this?
So if I'm able to understand your question, you are asking about the causality. What is causality assessment, right?
Yes, sir.
So causality assessment is the association between, means we establish association between the drug and the event. So there are two terminologies I would say first of all. So the two terminologies are like one is um, adverse drug event, and the second one is adverse drug reaction. So जहां पर हम बात कर रहे हैं एडवर्स ड्रग इवेंट की, तो यहाँ पर अभी कॉजलिटी रिलेशनशिप स्टैब्लिश नहीं किया गया है. बट व्हेन वी कम्स टू द रिएक्शन पार्ट, कि हाँ वहां पर एक कॉजलिटी असेसमेंट को हमने स्टैब्लिश किया है, तभी उसको हम एडीआर बोल रहे हैं. नाउ जब हम एडीआर की बात करते हैं तो यहाँ पर हमारे पास एक स्केल होता है. हम लोग अपने आप से नहीं बोल सकते कि नहीं इसने किया है या उसने किया है या इसने नहीं किया. हमारे पास एक पर्टिकुलर स्केल है. तो हम इंडियन इंडस्ट्री में, या कहें कि हम फार्माकोविजिलेंस प्रोग्राम ऑफ इंडिया में जो स्केल फॉलो कर रहे हैं, वो WHO कॉजालिटी असेसमेंट स्केल है, जिसमें हम सर्टेन, प्रोबेबल, पॉसिबल जो भी उसकी कॉजालिटी होगी वो स्टेब्लिश करेंगे, और उसके सेट पैरामीटर्स हैं. अगर वो उसमें उस सेट ऑफ क्वेश्चनर में फॉल करेगा तो हम उसको बोलेंगे ये सर्टेन है कि हाँ इस पर्टिकुलर ड्रग से प्रॉब्लम हुआ. अगर अगर हमारे को लग रहा है कि उसमें फॉल नहीं कर रहा है सर्टेन वाले पोर्शन में, देन वी कम्स टू द प्रोबेबल पार्ट. इन द प्रोबेबल पार्ट देयर इज अ सेट ऑफ क्वेश्चन. अगर वो उसमें फुलफिल हो रहे हैं तो हम उसको बोलेंगे कि हाँ ये प्रोबेबल है. तो ये एक पार्ट है कॉजलिटी असेसमेंट करने का. इसके अलावा भी देर आर अदर मेथोडोलॉजीज फ्रॉम विच वी डिफाइन द कॉजलिटी, लाइक नारंजोस कॉजलिटी असेसमेंट स्केल. तो वो भी एक स्केल है. अगर आप मैं मतलब यहाँ पर तो मैं अभी आपको नहीं दिखा पाऊंगा, बट अगर आप बोलेंगे तो मैं आई विल शेयर विद यू द causality assessment skills. So I hope कि मैंने आपको आंसर किया.
Okay, thank you, sir.
Well,
I would like to again thank you, sir, for sharing your knowledge regarding pharmacovigilance. Thank you so much, sir. Now, I would like to introduce our third speaker, Dr. Hitesh Kumar. Sir is recently working as an assistant professor in the Department of Botany in Sardar Patel University, Himachal Pradesh. Sir has more than 12 years of experience in teaching and research. His area of interest are plant molecular biology, gene isolation, and functional genomics. Sir has 4 patents and has a number of national and international publications. Beside that, Sir has traveled to the different nations to win numerous honors and recognition from professional association. He is a life member of many professional body and has received many fellowship awards. Sir is also a reviewer and editorial member of various scientific journals. Today, Sir is going to deliver his talk on herbal plant stevia. I request you, sir, to please enlighten us. Over to you, sir.
Thanks a lot, Miss Aman Patel, for your kind words. Am I audible?
You are audible. Yes, sir, you are audible.
Okay. Good afternoon, one and all. So, first of all, I want to thank the organizers for giving me this opportunity to be the part of this event. Also, I want to congratulate the patrons, convener Dr. Amit Sharma, technical advisor Dr. Amit Singh, organizing secretaries Dr. Meenakshi Sharma and Ms. Pratencha Sharma, and whole organizing team of Sri Sai University Palampur for organizing this wonderful event. Since I was told to speak on medicinal plants, so I chose the present topic to the audience of young postgraduate and research students to match with the present theme of this symposium. This is a study which I had performed in the recent past, and I hope it will give some insights to the young audience that how they can go for the molecular studies of a plant. So without any delay, I am starting my presentation now. So my screen is visible to all.
Yes, sir, it is visible.
Okay. So this is the topic for today's presentation. Molecular basis of steviocyte biosynthesis in Stevia ribaudiana botany, which is a source of non-calorific sweetener. So about this plant, a brief introduction. This plant belongs to family Astraceae, and it accumulates a cocktail of very sweet compounds, which are approximately 300 times sweeter than common table sugar. And these compounds, they are used as non-calorific sweeteners. So the country of origin of this plant is Paraguay, which is a South American country. However, in these days, this plant is grown in many countries around the world. So this plant is, since it is known for the stevia glycosides, so these are basically the derivatives of a diterpenoid alcohol. Uh, and the various steviol glycosides which are present in stevia are steviol bioside, stevioside, riboside, A to F, and then dalcoside. Of these major steviol glycoside, they are stevioside and riboside A. So this is the basic skeleton. Are my slides moving? Can you see the next slides, please? Can
Yes,
someone?
sir. Yes, sir.
Oh, right, right. So this is the structure you can see in the black color. This is uh, the uh, basic backbone, and R1 and R2 groups. They are basically the glycoside components, which varies in different steviol glycosides. Like in stevioside, the R1 position is occupied by a single beta glucose, and at R2 position there will be two glucose molecules which are linked by 2,1 linkage. So all these steviol glycosides, they vary at these R1 and R2 groups. Now, the biological and pharmaceutical properties which are associated with stevia, they are antihypertensive, mean it can used to cure high blood pressure. Anti-hyperglycemic, mean you can be used by diabetic persons for their treatment, and also as non-calorific sweetener. Third, it's immunomodulatory, means enhance your immune system. Antioxidant, having the properties to quench the free radicals. Anti-inflammatory and anti-cancerous properties also there, and antimicrobial properties, including antibacterial and antiviral properties. So now I'm coming to the major work which we have performed in the past. So this is regarding the steviol glycoside biosynthesis pathway, that how these compounds, they are synthesized in this plant. So this pathway, because these are terpenoid compounds, steviol glycosides, they are terpenoid compounds. So
Hello. So your voice is not audible. So your voice is not audible. Hello.
Madhu, you know.
So your voice is not audible.
Because of some technical issues, sir has left the meeting. He's joining very soon. So I request all the participants to please remain in the meeting.
sorry, I had lost my Wi-Fi connection. That's why it was there. So am I now?
Yes, sir, you are audible, but your voice is breaking.
Okay, okay. Actually, there is some problem in this Wi-Fi, so that's why it's ugly. Uh, Still, it's cracking, or
No, 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 sir. It's fine now.
okay, it's okay now. Okay, okay. My slides are visible.
Yes, sir, it's visible.
You can see that slides, I think, pathway slides, right?
Yes, sir.
Right. I was telling about this pathway of synthesis of these isopropyl units, dimethyl allyl phosphate and isopentenyl diphosphate.
Sir, your voice is
So this,
breaking.
occurring. Uh, can you reconfirm it, please? My voice is audible now. My voice is audible now. Okay, let me try it from my cell phone. I have connected it from my phone now.
Due to some technical issue, sir has left the meeting. Sir is joining very soon.
Yes, sorry for the inconvenience. Now my voice is audible, I hope.
Yes, sir.
Okay. So I'm on the stevia glycoside biosynthesis pathway screen, this slide, right?
Yes, sir.
Okay, okay. So after the synthesis of these isoprenoid units, then five more steps are there for the synthesis of this diterpenoid alcohol, which is called as steviol. And this action is carried out by certain these enzymes, this GGDPS, geranyl diphosphate synthesis, then copelyl diphosphate synthesis, corrin synthesis, and at the end, cornoic acid hydroxylase. And once this steviol is formed, then this steviol is converted into these different steviol glycosides via these glucosyl transferases. Are my slides moving? Can you confirm, please?
Yes, sir. Yes, sir.
Okay. So now I'm coming to the importance of this work, because I was telling in the beginning that this is the very basic work, and the research students, and especially the postgraduate students, they can get some insights that how they can go for the research work in a medicinally important plant. So in the beginning, the molecular data for stevia was very limited. Very few genes they were reported, and moreover, there was no comprehensive picture that how these compounds they are synthesized at molecular level. So we tried to get some insights from this present work. So the objectives which were fixed in the beginning, they were cloning the genes which are involved in steviol glycoside biosynthesis pathway, and secondly it was to understand the regulation, how these genes they are regulated at molecular level, because these compounds they are synthesized in very high concentrations. They are accumulated up to 30% of the dry weight of the plant. So how these compounds they are synthesized, so we were interested to know about it. So the methodology which was designed in the beginning for cloning, we followed very basic approach. We designed these degenerate primers, and then we cloned the full length genes using these partial sequences which we cloned using these degenerate primers. And for regulation studies we carried two-way approach. First was through the construction and screening of genome Walker libraries to clone the promoters. And second was through expression analysis, which was carried out through this semi-quantitative RT-PCR. So, because stevia is known for high accumulation of these steviocytes, so the commercial methods which were available already, they were not able to isolate RNA properly from stevia. So we designed a in-house reagent kit to isolate RNA from stevia. So we later patented this method also. This was designed by our own team, and it was later also published. So you can see the publication detail at the bottom of this slide. The title of this work is an RNA isolation method to isolate RNA from tissues which are high in scanty metabolites. So the method we developed, it was able to isolate RNA from different parts of the plant, leaf, stem, and root to a very high quality and quantity. You can see here the distinct bands of RNA, which shows that these bands show that the RNA is of very high quality, because there is no smear there around the bands. So after isolating RNA, we prepared cDNA from that RNA, and went for the cloning of these genes which are involved in steviol glycoside biosynthesis. So we used different softwares to design the primers. Uh, First of all, since these genes they were not reported from stevia, so we took uh, the homologue from other plants, then go for blast search. Blast, you might have known about this program. This is basic local alignment search tool. So we aligned different genes from that database, and we found the conserved regions which are present in these genes. And using these conserved regions, we design the primers. So in this slide, you can see the amplification, partial amplification of the cDNAs of different genes which are involved in steviol glycoside biosynthesis. So these were the partial gene sequences. You can see here, these are not full length genes. These were only partial sequences. So once we sequenced these partial sequences, then we went for the full length amplification. So for full length amplification, we used a technique called RACE, RACE. So this RACE is, you can say, this is random amplification of cDNA ends, because the partial genes, they were lacking their 5' and 3' end. So we designed gene specific primers and adapter specific primers to clone the 5' as well as 3' regions of the partial sequences. So in this slide, you can see here the uh, DNA gels, which are of 5' and 3' uh, gene sequences of the various genes of SC biosynthesis pathway. Once we amplified these genes, then we cloned these in a cloning vector and sequenced these sequences. So after sequencing these 5' and 3' regions, we overlapped them and designed primer from start and stop codons of the genes, and used these primers to amplify the full length gene sequences using the cDNA of stevia. So you can see here in this slide that these are the gel pictures of different genes which were cloned to full length. And also in this slide, you can see the number of base pairs which one gene contains. So after sequencing them, I have submitted them to this NCVI Gene Bank, National Center for Biotechnology Information. And at the right column, you can see here the accession numbers of these gene sequences. After cloning full-length genes of steviol glycoside biosynthesis pathway, I went for the functional validation of these genes. So functional validation was carried out by a two-pronged approach. First was through bioinformatic analysis, in silico analysis using computers, various computer programs. And second was through complementation assays. In this approach, we used these knockout mutants of bacteria. So using first approach of this in silico analysis, we were able to identify the various characteristic domains which are found in the enzymes of these non-malonate pathway and downstream enzymes of steviol glycoside biosynthesis pathway. So the characteristic domains which are characteristic of each enzyme, they were also visible in these genes which we have cloned. So you can see here the phylogenetic tree. We have performed phylogenetic tree analysis for each gene, and the genes, they were closely related with that of plant, since it was from stevia, which is a plant. So they were closely related with that of plant, and their and these bacterial counterpart, they were far apart in the phylogenetic tree. So I'm skipping these files and these slides. So after this in silico analysis, functional validation, as I told earlier, it was through complementation assays in the bacterial mutants. So a mutant, this is basically lacking certain gene, and these mutants, they can grow on certain selective media. Here in case of in this slide, you can see here at the left bottom, the bacteria, they were able to grow in the presence of MVA, means malvolate. Once they are supplemented with malvolate, they were able to grow. However, if you don't apply malvolate to the media, then the bacteria were not able to grow. You can see it in the left top right corner without malvolate. When we transform these mutant bacteria with the genes which we have isolated and kept these genes inside a expression vector, so you can see at the top right corner that the bacteria, they were able to grow when they were transformed with the gene which was isolated from stevia. So this is how we prove that our gene, it uh, express a functional protein, means enzyme, which was able to make these mutant functional. So you can see here the negative control in which we used only this expression vector. The gene was not put inside that vector. So the bacteria, they were again, they were not able to grow on the media without malvolate. So the right panel of this photograph, it is without malvolate, and left panel is with malvolate. So this is how this knockout approach is used for functional validation of the genes. And this stain we have procured from an international lab of Tomohisa Kozuyama in University of Tokyo, Japan. Second complementation assay was for MDS gene of stevia. So here you can see again, when we use glucose in the medium, the mutants, they were not able to grow. However, when arabinose was used as media, the mutant stains, they were able to grow. However, when we transformed again these mutants with the gene of stevia, so these you can see here at the right bottom, that when the bacteria, they were transformed with MDS gene of stevia, they were able to grow even in the absence of arabinose. So that's why we proved that this gene encode a functional enzyme which is required for the synthesis of intermediates which are used in non-malvolate path. And this is complementation assay for another gene, this HDR gene of stevia. So again, here you can see in presence of arabinose, these mutants, they can grow. However, when they are complemented with the gene of stevia, then they were able to grow even in the absence of arabinose. So this strain was procured from Edinburgh, Scotland, by Dr. Millicent Masters. She has prepared this knockout mutant. So these three genes, they were functionally validated in this way. After functional validation, we went for these regulation studies. So for this, we cloned the promoter sequences of the genes. So for this, what we do, we prepared genome walker libraries. You can see here the diagrammatic presentation. So we took the DNA of stevia and digested it with the different restriction enzymes. 4 restriction enzymes, they were used: DRAW1, EcoRV, PVO2, and STU1. So after digesting them in separate tubes, we ligated the adapter sequences to these fragments which were generated through this restriction enzyme digestion. And after ligating the adapters, we used two types of primers here. One was adapter specific primer, and second was gene specific primer. So in some cases, we got amplifications in the primary PCR. However, in cases in which we didn't get amplification, we went for nested PCR using this adapter specific primer and gene specific primer, which were prepared from the nested positions in the gene. So you can see here the these upstream regulatory sequences, which were cloned for different genes, and their base pair length is also given in the second column. And uh, they were also submitted to gene bank, and the accession numbers which are allotted to these regulatory sequences, you can see here at the right column at the extreme right. So you can see here the DNA gel picture of different amplifications which we got using these genome walker libraries. So these bands, they were excised and cloned and sequenced, and the sequence I have addressed earlier, these are the same sequences which we cloned. So once we cloned these promoter sequences, then we went for in silico analysis using various bioinformatic tools. So when we perform this in silico analysis, you can see here in red color, I have highlighted them in red color. So these are various motifs which are found in these promoter sequences. So you know that promoter is used for the transcription of a gene. So various controls are there for the regulation of transcription. So the controls which were you can see here different motifs. You can see here different motifs which are present. So different transcription factors they use to bind to these motifs. So again, I am skipping these slides. So now I will give a brief idea that which kind of regulatory elements they were present in these promoters of stevia glycoside biosynthesis pathway genes. So there were light responsive elements, low temperature drought responsive elements, phytohormone responsive elements, including auxin, abscisic acid, gibberellins, cytokinin, and then signal transduction elements were also there, and tissue specific regulation elements were also there. So I will discuss them one by one. Light responsive elements, you can see here the motif, name, and sequence which was found in this light responsive element. And at the right column, you can see the name of gene in which these light responsive elements were found. I will later know you out the implication of these elements and how they are related to the expression pattern of different genes. So there were also low temperature and drought responsive elements. You can see here the name of motif and the consensus sequence and the genes in which it was found. Phytohormone responsive elements, you can see here in table. Responsive elements, you can see here in the table. In element, then elements, these and specific elements expression studies we did about gene expression, that how genes molecular in stevia. So in this in the left side, we have got the gel picture of semi-quantitative PCR, which we have performed. We isolated RNA, then prepared cDNA, and used an internal control. In this case, it was 26S rRNA gene. You can see here that in root, stem, and leaf, the 26S rRNA gene, it is equally expressed in all parts. However, when we went for these tibial glycoside biosynthesis pathway genes, then we can see here that below leaf, there is higher expression in case of leaf. However, an intermediate expression, it was noticed in green stems of stevia, and expression was least in case of roots. So when we went for content analysis, we performed the content analysis through HPLC, high performance liquid chromatography. So you can see here at the right bottom, the steviol glycoside content analysis. We quantitated these two stereoglycoside. One was stereoside and second was riboside A. So you can see here the content, they are very high in leaf, and they are in intermediate quantity in stem, and root contains least amount of these stereoglycosides. So what we do, we perform correlation analysis. So in correlation analysis, we studied the correlation of the expression of different genes with that of the cereal glycoside contents. So there was a very high correlation, which showed that the genes, they are involved in cereal glycoside biosynthesis pathway. So similarly, we perform this gene expression in different developmental stages of leaf. You can see we sampled the leaves from first, second, third, fourth, and fifth node positions of stevia, and we studied the content. You can see in the right side, these are the contents, stereoside and riboside. So the steviol glycosides very high in third positions, and they then decrease in fourth and fifth leaf. See, first and second leaf, they also contain lesser amount of these. So when we studied again this correlation analysis with that of the gene expression, so you can see here two genes, three genes, they are highlighted in red. This DXR, KO, and NCYPR. This NCYPR is a cytochrome P450 reductase enzyme. So these three genes, they showed correlation with that of the stereoglycoside contents. Means you can see that there are pathways, and there are certain regulatory genes in these pathways. So here we identified these regulatory genes in case of stereoglycoside biosynthesis pathway. So this is correlation for riboside content. The previous was with that of stereoside content. So once we identified that light is playing a role in stereoglycoside, so we perform certain experiments at natural conditions, and we also did them in these plant means in enclosed conditions in these polyhouses, etc. So you can see here in the right side, when plants, they were exposed to continuous dark conditions, so expression was very low for this ST biosynthesis. Okay? However, when they exposed to light conditions, then the expression it was very high. So we also did experiments for by applying external cues like light dark conditions. We kept plant for continuous light conditions, for dark conditions, and one was for control. So you can see here again the plants, when they were exposed to light conditions, the genes of HG's biosynthesis pathway, they showed higher expression as compared to dark conditions. So these were the expression in response to gibberellin treatment. We also performed treatments. We to the cuttings, we applied these gibberellins. So you can see expression of the genes, different genes in response to gibberellins. This is in response to this kinetins, and this gene expression pattern is for methyl jasmonate treatment. So this was the status at the end of this work. You can see here all the genes of stereoglycoside biosynthesis pathway, they were cloned, and their upstream regulatory sequences, they were also cloned. So this pathway is complete now. So these are the publications, different publications from this work, and the work which was continued after this. Recently, I had prepared the next generation sequencing libraries of stevia, and approximately 18,000 unigenes, they have been cloned from stevia in my recent work, and these are the international labs you can see here from Canada, Scotland, and Japan, and this work was carried out at CSIR ISPT Palampur, and fellowships, they were from CSIR, ICMR and CSIR, they provide funding for this research work. So thank you, and I'm ready to take the questions if there are from the audience.
Thank you, sir. Thank you so much for enriching us with the knowledge on stevia. Now we have some questions. The very first question is, can we completely replace sugar with stevia?
Yes, as I told in the beginning that this plant, it contains non-calorific sweeteners, and even you can see nowadays Diet Coke and Diet Pepsi, they are using these compounds in their preparations in place of this sugar. So because there are certain artificial sweeteners are also there, so these artificial sweeteners they are very long. So that's why some these sweeteners in is option these days.
Thank you, sir. The second question is it safe to give stevia to diabetic patient?
Yes, because diabetic stevia, since it's non-calorific, you can give it to diabetics. And besides it is also found that it is very useful for type 2 diabetics, which are dependent on these drugs. So not for type 1, insulin dependent, only for those who are taking medicine. So it has been shown that stevia, steviol glycosides, they are helpful in reducing the blood sugar levels also. So it has pharmaceutical value. Besides non-calorific, it has pharmaceutical values also in reducing blood sugar levels.
Okay. Thank you, sir. It will be a great pleasure to listen you again in our future events. Thank you, sir.
Thank you, madam.